Take a deep breath in and out. What do you breathe in that keeps you alive? The answer seems obvious. We all take it for granted. It's all around us. It's oxygen. But after you've breathed the oxygen in, where does the oxygen go? When I was young, my mother read me a story about how oxygen keeps you alive. Today, I want to tell you another story, beginning with a fundamental scientific question of where the oxygen goes. Unexpectedly, this leads to a rare, lethal, pediatric neurological disease, and to the prospect of the first treatments for this disease. In order to follow oxygen, we need to know first that there is such a thing as oxygen. To do this, we need to go back 300 years to the discovery of oxygen by Carl Scheele and Joseph Priestley, and to the discovery by Antoine and Marianne Lavoisier that oxygen is critical for combustion and for respiration. Scheele and Priestley were the first two scientists to isolate oxygen and show that it is part of our atmosphere. Lavoisier showed not only that oxygen was important for combustion and respiration, but also named it. Lavoisier showed that a guinea pig, a living organism, and a fire that produced the same amount of heat consumed the same amount of oxygen. From this, he concluded that respiration was a slow fire. And 300 years later, we know that he was right. By taking careful, precise, quantitative measurements, Lavoisier showed that we use oxygen to keep the fire of life burning. All three of these scientists were interested in knowing where the oxygen went, and Lavoisier actually made tremendous progress doing so. But none of them could actually answer the question of where oxygen went because none of them had the tools to do so. 300 years later, our lab asked the same question: where does the oxygen go? In order to do this, we thought it would be wonderful to have a little flag that we could put on the oxygen to follow it around the cell, in much the same way that putting a pennant or a flag on a bicycle allows you to see it much more clearly. Luckily, we have a way of doing this by treating cells with gaseous heavy oxygen, or 18O2. Each atom of 18O2 is 2 neutrons heavier than the 16O2 that all of us are breathing right now. The difference in mass between an atom of 18O2 and of 16O2 is a billionth of a billionth of a billionth of a kilogram. But measuring that tiny difference is no problem for an instrument called a mass spectrometer. We used 18O labeling followed by mass spectrometry to figure where the oxygen goes. Once we had done so, it became clear that there was one critical molecule that took up 18O more than any other. This molecule turned out to be a long-lost precursor in the synthesis of another critical molecule called coenzyme Q10, or CoQ10. CoQ10 is found in every single cell in our body. Children who cannot make CoQ10 develop a rare and lethal pediatric neurodegenerative disease. This disease consists of a number of symptoms: seizures, spasticity, or tense, tight, paralyzed muscles, and ultimately culminates in the death of these children by the age of 8 or 9, if they make it that long. How could the loss of such a simple molecule or a single molecule cause such a catastrophic disease? It turns out that you need CoQ10 for producing energy, and your brain needs a lot of energy to think. The loss of CoQ10 in children inevitably causes neurological symptoms. We thought that we knew everything about CoQ10, but it turns out that we breathe oxygen not just to live, but to build the very molecules that enable us to survive. You might be asking, haven't I seen CoQ10 before? And indeed, you have. CoQ10 is found on the shelves of every pharmacy and supplement store, and many of us take it as a supplement. So if these children can't make CoQ10, and you can buy CoQ10 in a pharmacy, why can't we just treat these children with CoQ10? This has been tried, but unfortunately, none of these children respond to very high doses of CoQ10. It turns out that CoQ10 is actually not very well absorbed by the body. But what if we could treat these children with the building blocks, the precursors of CoQ10 that our lab discovered? At this point, our lab set off into an unknown area of research for us. It became very clear that to understand and to develop treatments for this disease, we would need models of this disease. We would need a mouse that mimicked the symptoms of this disease. We would ideally need a mouse that could not build the building blocks of CoQ10 and would recapitulate the symptoms that we find in the patients. So, like Lavoisier, we needed a model. Science changes, but the scientific method remains the same. We obtained mice that could not make the building blocks of CoQ10. And just like the patients, they developed seizures, spasticity, and none of them lived past the age of 2 weeks. We then treated them with CoQ10, and nothing happened. Then we treated them with the building blocks of CoQ10 that we had discovered. Within 2 days, the first mice were standing on all 4 paws. A month later, they were walking. It was a miracle, and at first, none of us believed it. But as we treated mice, and with additional cohorts of mice, and we treated more and more mice, they lived longer and longer. And now we have mice that live a full life for a mouse that have been treated with these CoQ10 building blocks. I am very excited to tell all of you that we have obtained approval to treat the first patient with this rare, lethal neurologic disease with the CoQ10 building blocks that we have discovered. Treatment is currently underway. We are currently very, very hopeful about the results. But more importantly, this treatment, this prospective treatment, has brought hope to the patient and to their family. When I started my lab, I wanted to make basic discoveries that were directly relevant to patients. I'm here to tell you that life-changing medical discoveries emerge by following the scientific method and the scientific data wherever they may lead, especially when they take you out of your comfort zone. If you want to really move the needle in patient care, you have to understand biology at a fundamental level. Our discovery emerged from a question first asked 300 years ago by scientists who did not know where their discovery would lead, but who were passionate about finding out the nature of the world around them. That question and their discoveries 300 years ago have led today to a prospective treatment for a rare and lethal neurologic disease. We did not know where we were going when we began this story. But this story and our discoveries have been more beautiful and more meaningful than anything we could possibly have imagined. Thank you.